Okay, so welcome to this next video in which we are studying the effect of sympathetic stimulation on the heart, and we're specifically now looking at how we increase the rate at which the heart beats, i.e. by how in the way in which we produce a positive chronotropic effect. Okay, right, so we have said that if we can increase the conductance of these HCN channels, then we're in business, because the gradient of this initial depolarizing portion of the action potential in the SA node cells will be greater, and that will mean that the length of the overall action potential will be shorter, so you can pack more of them into a certain amount of time, for instance 60 seconds, a minute. So you'll get a greater number of action potentials being fired in a minute, and since each one of these action potentials is going to produce a beat of the heart, you're going to get a greater heartbeat. Okay, right, so what we want to do is now discuss how stimulation of the beta-1 receptor is going to trigger this. So let's just have a quick reminder of what happens when we stimulate the beta-1 adrenergic receptor. So here's our cell membrane, let's say, of our cardiomyocyte. Here is our beta-1 adrenergic receptor, this 7-transmembrane receptor. Okay, and we know now that it's coupled to the GS G protein. So here's the GS heterotrimeric G protein with the alpha, the beta, and the gamma subunit. And we know that this is going to go on and activate the adenylyl cyclase 5/6 enzyme, which is here. Okay, so this adenylyl cyclase 5/6 enzyme, the AC5/6, is then going to produce cyclic AMP. And in fact, we do not even need to look at protein kinase A for this. So it's the cyclic AMP that is going to activate the HCN channel. So when noradrenaline comes along, it will bind to beta-1 adrenergic receptors which are on the sinoatrial node cells. Okay, and I'll draw these beta-1 adrenergic receptors in pink here. So these are in this violet purple color. Okay, we know that the beta-1 will activate the alpha-S subunit here of the heterotrimeric GS-G protein, okay, which we'll draw in turquoise here. We know then that the alpha-S subunit with the GTP bound to it will then activate the adenylyl cyclase 5/6, which we'll do in blue here. And again, adenylyl cyclase 5/6 means either adenylyl cyclase 5 or adenylyl cyclase 6. It does not refer to some bizarre chimera. Okay, and then that will produce a cyclic AMP. So cyclic AMP is going to go up in our sinoatrial node cell. Cyclic AMP binds to the HCN channel. Now, in order to understand this a bit more, we need to look at the structure of one of these individual HCN proteins. So you remember me telling you that the HCN channel consists of these 4 HCN proteins tetramized together to form this channel. Now, what we're going to now do is take out one of these proteins here and look at its membrane spanning structure. So if this is the phospholipid bilayer, its structure is like so. Here's the amino terminus of the polypeptide. It spans the membrane once, it spans the membrane again, it spans it a 3rd time, a 4th time, a 5th time. It tries and fails, and then it spans it a 6th time. So it then has a really important domain here. Okay, and then finally it finishes with its carboxyl terminus down here. Okay, so this is the membrane spanning structure of a single HCN protein. So 4 of these join together to make the HCN channel. Okay, right. Now, the important thing is this domain here in orange. This is what's known as the nucleotide binding domain. Okay, so this is a nucleotide binding domain. And this is where cyclic AMP will bind to. So that's the nucleotide binding domain. Okay, so when cyclic AMP goes up in the cell because of the activation of the beta-1 adrenergic receptors, the cyclic AMP is going to come and bind to this nucleotide binding domain, and it's going to activate the HCN protein. So you'll get one cyclic AMP binding to the nucleotide binding domain of each one of the 4 HCN channel proteins which are making up the HCN channel, and when that happens, it will cause a conformational change in the HCN channel proteins, which will increase the conductance of these HCN channels. So, when the HCN channels open upon hyperpolarization of the cell membrane electrical potential difference to -70 millivolts, then they will open, and the rate at which sodium ions will move through that pore into the cell will be greater. So you will depolarize the cell membrane quicker, so you'll get to -60 millivolts quicker, and then the T-type voltage-gated calcium channels will take over, and you'll get the rest of the action potential occurring. Now, if this initial depolarization is steeper, then the overall time interval for one of these action potentials is shorter, so the number that you can fit in a minute is greater, and therefore the sinoatrial node is going to be firing these action potentials spontaneously at a greater rate, and that's going to lead to a greater heart rate, a greater greater amount of times that the heart beats in a minute, which will mean that it overall expels more blood. So overall, when you stimulate the heart with the sympathetic nervous system, you're going to increase the heart rate. So you're going to increase the number of times the heart beats in a minute. In addition, you're going to increase the force with which the heart contracts. So on each individual heartbeat, the heart will expel a greater fraction of the blood volume that is actually within it. So that also is going to help you increase the cardiac output. So overall, the amount of blood the heart moves from the venous side to the arterial side in a minute is going to go up, and that's going to help you fight or flight.